Hello my friends. In this video we will start chapter 3, Biological Macromolecules. This is basically the meat and potatoes of biology. We discussed in the last two chapters, the first chapter being basically what is biology, and chapter 2, what is biology made out of. Chemistry is the smallest, is really the smallest you can go in terms of biology. However, we were talking about chapter 3, which is micromolecules. This is basically like, I guess, the building blocks of biology. And in this video, we will be discussing two parts of the chapter. Chapter 3.1 will be the synthesis of biological macromolecules, which is essentially an introduction and is very short. Then we will go into carbohydrates. So let's get going. First, as you learned, biological macromolecules are large molecules necessary for life that are built from smaller organic molecules. In other words, a macromolecule would be like a nucleic acid. That would be the monomer, which many of them will combine into making a polymer known as DNA, which we'll discuss in 3.5. So now you kind of have an idea. A monomer is one macromolecule in essence, while a polymer is the combination of macromolecules. And carbs, we will discuss that here shortly. Now, in terms of how polymers and monomers are formed and how macromolecules are combined, there are two processes. The first one is the dehydration synthesis. In essence, dehydration synthesis is when two independent monomers, which are single subunits or building blocks of macromolecules, when two or more monomers combined, they release water, hence dehydration. So here you see before they're combined, they have on the ends of their chains two HO molecules, or OH, however you want to say it. Then when they combine, the oxygen molecules one oxygen molecule combines to link the two chains together, and it releases the two H's and the one O, water. So dehydration synthesis is when water is released so the chains can combine. In contrast, when it's time for the chains to be broken up, it's called a hydrolysis reaction. In other words, when water is added to the system, it breaks these two up and adds OH to both. Well, OH to one and then H to the other, forming OH. So, to recap, just real shortly, dehydration synthesis is two chains combining and releasing water, and hydrolysis is water being added so two monomers can be broken up. Alright? Great. And of course, when it's combined, these things are known as polymers. Alright, now specifically let's get into carbohydrates. Most people are familiar with carbohydrates, one type of macromolecule, especially when it comes to what we eat. To lose weight, some individuals adhere to low-carb diets. Athletes, in contrast, often carb load before important competition to ensure they have enough energy to compete at a high level. A simple sugar is a compound that is a component of starch and an ingredient in many staple foods. Carbohydrates also have other important functions in humans, animals, and plants. The formula basically for a molecule like this is CH2O, N subunit, where the N is a number of carbons in the molecule represents carbohydrates, hence carbs. It has carbon atoms and hydrogen atoms. The number and structure differ depending on the sugar, and we'll get to that in a minute. The monomer, if you will, is the monosaccharides, or sacharides, however you want to pronounce it, which are simple sugars, the most common of which is glucose. Many of you are familiar with that. The number of carbons usually ranges from 3 to 7, and it really depends on what a sugar it is. Most monosaccharides names end with the suffix "-ose, such as glucose, fructose, saccharose, etc. If the sugar has an aldehyde group, the function group with the structure R, which would be the R group, CHO, it is an aldose. And if it is a ketone group, the function with it has RC double O, R. In other words, two R groups sandwiching the carbon and double double what's it called? Double bond O. It is a ketose. So you get the names. And of course, go by numbers. If it has 3 carbons, tri, 5 pent, and 6 hex. You get the idea. And here are some of them right here. You can tell it being a carb because it has carbon and a double. Generally, you can tell it's a carb by 3 functions. By its carbon, its double bonded oxygen to a carbon, and the presence of H, generally with OH as well. In fact, I think all of these listed here have OH, and that's due to the fact that it's not combined. So, generally they were combined with some OH, either with their own OH or with another OH on this side. You get the idea. Alright. The chemical formula for glucose is C6H12O6, and often it will be reduced to CH2O, but we're not worried about it right now. In humans, glucose is an important source of energy. During cellular respiration, breathing, energy releases from glucose, and that energy helps make adenosine triphosphate, ATP. You are going to want to remember ATP because pretty much all of life functions around the production and release of ATP. So remember that. ATP is important. ADP is important as well. And here are some of the probably most famous simple sugars. The glucose. You can see here it's a carbon chain with 4 H's on one side, 1 OH, 4 OH's on one side, and 1 H, with a tail being an H. But I think all these have tails being an H as well. Glucose is, galactose, Super Mario Galactose, if you will, tip your waiter, is very similar to glucose, but the sequence of OH and H is slightly different. And of course, fructose is a lot different because it's a double bonded O is down here a little, is a little lower on the chain normally than most other chains. It's actually quite remarkable how much these three are different, simply because of a slight variation in not genetic makeup because they're using the same atoms, but in structure. But yeah, you can just change structure and everything changes. In fact, when we get into genetic mutations, like way down the road, you'll see how a tiny change can do so much. Alright. Oh, okay, try to read this. Monosaccharides can exist as a linear chain or as a ring-shaped molecule. In aqueous solutions, they are usually in ring forms, which we will see down here. Glucose is a ring form. In a ring form can have 2 different hydroxyl arrangements, hydroxyl being anything with OH, around an anoramic carbon 1 that becomes asymmetric in a ring formation process. If the hydroxyl group is below carbon number 1 in the sugar, it is the alpha position. If it is above the plane, it is a beta position. So let's look at this right here. It's in a ring shape. Every little corner means carbon. And believe it or not, that took me a while to learn. I never understood, like, how is this connected? Was because if there's no letter here, it means each little corner is a lone carbon atom. Everything else that's not a lone carbon atom is lifted. Like, for instance, we just saw up here on glucose that here the O is double bonded to carbon, and you don't see the C here because each corner of the ring is carbon. It's best to remember that. If you don't see carbon, it's simply because each corner of the shape. Here you see 1, 2, 3, 4, 5 corners of the 6-corner thing. Does not have a letter. It means that it's 5 carbons here. And we can, okay, what is this? This is glucose. So hold on. Yeah, glucose. And we can verify this up here. It has 1, 2, 3, 4, 5, 6. Oh, that's right. I'm sorry. This is what we're looking at, not this. Because this is just how it's looking. So sorry. 1, 2, 3, 4, 5, 6. Well, for some reason the art here is a little off. There should be a corner here, but basically there are 6 carbons, like it really should be. So that's a little weird. Ribose and fructose are right here as well. Here it's 1, 2, 3, 4, and then 5 because it's a corner. Alright, now we get into disaccharides. Disaccharides, as you know, di means 2, form when 2 monosaccharides undergo a dehydration reaction, which we discussed earlier, was when 2 monomers lose water and combine and release water. During this process, one monosaccharide's hydroxyl group combines with another monosaccharide's hydrogen, releasing a water molecule from a covalent bond. If you recall, a covalent bond if you guys recall, a covalent bond is when 2 or more atoms share electrons. For instance, share electrons to fill their valence shell. For instance, hydrogen often combined with carbon, so the hydrogen can share one atom to fill its valence shell. Carbons can share up to 4 hydrogen atoms to fill its valence shell. Scientists call this, which is the forming of a covalent bond within disaccharides, a glycosidic bond. Glycosidic bonds can be in alpha-beta type, just like what we discussed earlier. An alpha bond is formed when the OH group is on the carbon 1 of the first glucose is below the ring plate. The beta is formed when the OH group is above the ring plate, like we see right here. Alpha, the OH, is below the ring plate. See it right here? And then the OH here, this in beta, is above. See it? Alright. And here we see the dehydration reaction. We have the 2 chains here with its 6 carbons, 1, 2, 3, 4, 5, 6. I forgot. You have to when you're counting the carbons, you also have to count the one that's shown. I didn't do that earlier. My apologies. Anyway, it goes through a dehydration synthesis and release water. Pretend right here it says plus H2O. And here's your glycosidic bond. The O is basically pulling and holding these 2 together at the carbons. Common disaccharides include lactose, maltose, and sucrose. Obviously, sucrose is the most common, which is table sugar. Yeah, your table sugar is a disaccharide. Alright. And here are the formations of each. You have your maltose, your lactose, and your sucrose. Now, at first glance, these look kind of the same, but they are different. Like I said, subtle differences. Like for instance, if we look at lactose and sucrose, sucrose has at the ends here 2 atoms, or in this case, not 2 atoms, 2 structures, whether it be atoms or molecules. And with lactose, only 1 on the ends here. The ends being the opposite sides of where the bond is. That's written the general shape is different as well. And then we got polysaccharides. Polysaccharides are basically long chains of monosaccharides, like it says here, and they're all linked. Each link here is combined by the glycosidic bonds, which is hard to say. Which again is basically what forms when going through a dehydration synthesis. The chain may be branched or unbranched, which we will discuss later. And it may contain different types of monosaccharides. Plants store starch in the form of sugars. In plants, amylose and amylopectic mixture, both glucose polymers, comprise these sugars in plants. We break these, basically, starch is their way of holding sugar. We hold sugar as carbs, or we use sugars as carbs, hold as glucose and stuff like that. Glucose starch comprises of monomers that are joined by alpha 1,4, alpha 1,6 glycosidic bonds, which is hard to say. Alright, actually, no, let's read this. Let's continue reading this. The starch in the seed provides food for the embryo as it germinates and can also act as a food source for humans and animals. Enzymes break down the starch that humans consume. For example, an amylase present in saliva catalyzes or breaks down the starch into smaller molecules, such as maltose or glucose. The cells can then absorb the glucose. Alright, here is two types of polysaccharides. Here is amylose, and here is amylopectin. And you can see here each one is combined into a is linked with each O's. I'm not sure why this one is separated. I think it's only separated to let you know what each individual one looks like, but I'm not entirely sure what these brackets are for. Okay. Oh, sorry, glycogen. That's why I couldn't say earlier. While plants store carbs as starch, we store as glycogen. Glycogen is in the animal equivalent of starch and is highly branched molecules used to store in liver and muscle cells. Whenever blood glucose levels decrease, glycogen breaks down and releases glucose in a process called glycogenosis. And cellulose is for plant cell walls. That's really different than starch. Starch is inside the plant and uses energy reserved. Cellulose is outside the plant and uses more of defense. I believe glycogen is also stored in fat in animals, but I think it's a little different. So I think we'll discuss fat at another point. Not sure in this video or not. Actually, no, we don't, because I think fats fall under lipids. So we'll worry about that later. And here's the cellulose fiber and what the cellulose looks if you look at an electron light. This is basically what you would look like if you look under a powerful microscope. And then for really small things, you can see this in an electron microscope, I think. Don't call me. The N here, I guess, represents how many past this point would represent. So if it's one here, 100, and 100 more, etc., etc. Alright. Carbohydrates serve various functions in different animals. Arthropods, such as insects, crustaceans, and others. I think what else falls under arthropods? Oh, crud. I just went over this with someone else. I don't remember. But arthropods is the most abundant, if we discuss like way down here, arthropods is the most abundant animal genus, I think. So we'll get there. But the exoskeleton of these animals is made of chitin. Chitin is also a major component in fungi cell walls. And now we discuss the benefits of carbohydrates. Are carbohydrates good for you? Some people believe that carbohydrates are bad, and that they should avoid them. Some diets completely forbid carbohydrate consumption, claiming that a low-carbohydrate diet helps people to lose weight faster. However, carbohydrates have been an important part of the human diet for thousands of years. Artifacts from ancient civilizations show the presence of wheat, rice, and corn in our ancestors' storage areas. Essentially, carbohydrates are important because, unlike stored fats, carbohydrates is instant energy. If you need to get work done, you need carbohydrates. However, if you don't really live an active lifestyle or don't really need a whole lot of instant energies, then carbohydrates could probably be avoided to a certain extent, and probably should be if you're trying to lose weight, because very often when you're trying to lower your diabetes chance or lose weight, it's the carbs you have to worry about more than anything, because carbs turn to sugar, and those carbs will affect you way more than any table sugar or sugar on the food labels ever will. So, but my expertise is not in dieting, so most definitely do not take my word for it on that. I really don't take my word for it here on anything. I'm simply giving a ASMR experience while teaching you about my job, or my soon-to-be job, I hope. So anyway, next video we will talk about is lipids, which, if memory serves, yeah, right here, is fats and oils. Fats, of course, being, I believe, the fat is energy reserve for animals, and oils energy reserve for plants. Even though I think that's not quite right, because I thought starch was, yeah, I just said starch was the energy storage for plants. But either way, oil is important for plants, and fat, as you know, is important for humans to a certain extent. So we will discuss lipids in the next video. So I just want to thank you all very much for watching. Have a wonderful day, and God bless.